Welcome to the last and final episode of Science with Jake, spoken with a sad heart because it's the last episode. Although who knows, maybe I'll make more episodes on my own because this has been kind of fun. In this episode, we're going to talk about synapses, what they are, the different types, as well as the types of receptors that neurotransmitters can bind to. First and foremost, what's a synapse? A synapse is a junction between two neurons or between a neuron and a muscle cell or a gland where the electrical signal is transmitted from what they call the presynaptic terminal and the postsynaptic terminal. That is the first neuron that is sending the signal, and the second neuron is the postsynaptic. Pre, beginning, post, after. Synapses can be either chemical or electrical, but most neurons in the nervous system communicate through chemical synapses. Let's zoom in and take a look at these different synapses. Cue the green screen in 3, 2, 1. On the left here, we have an electrical synapse, and on the right, we have a chemical synapse. Electrical synapses differ from chemical synapses in that they have a physical, direct connection between them. You can see that right here with the gap junctions that are connecting them right there. This allows electrical synapses to provide bidirectional communication, meaning it goes both ways, as well as faster communication, or at least faster communication than chemical synapses. Chemical synapses only allow communication in one direction, so they're called unidirectional. And as you can see, they have a completely different method of action. They're using these things called neurotransmitters, which are basically the chemical messengers of the nervous system. Note that both synapses, though, are activated by an action potential reaching them. However, when an action potential reaches the presynaptic terminal of the chemical synapse, something different happens. You can't see it in the diagram here, but in that little section where the neurotransmitter, those little black dots are being released, that is caused by the opening of voltage-gated calcium ion channels. When the action potential reaches the end of that presynaptic terminal, those calcium ion channels open up, calcium rushes in, and the neurotransmitter is triggered to release. As you can see, the neurotransmitter molecules are packaged in little vesicles, which are those little circles in the presynaptic terminal here. And as they're released, they just get sent out into the synapse where they then go and bind to these different types of receptors. These membrane-bound receptors are found in the dendrites of the next neuron, and they can be either ionotropic or metabotropic, as you can see in the diagram right here. The two receptors we have on the right there, those are the ionotropic receptors, and those are basically just ion channels that open in response to the neurotransmitter binding. On the left, however, we have the metabotropic receptor, which is also known as a G-protein coupled receptor. When neurotransmitters bind to the G-protein coupled receptors, it can also result in the opening or closing of ion channels. However, it is done through a series or a cascade of reactions. This triggered chain of events involves these chemicals called second messengers, which basically go and activate other things. So one thing activates this, which activates that, which could activate two other things, and it creates a large amount of diversity in signal amplification. So even though a chemical synapse is slower than a physical electrical synapse, it allows for a more diverse array of communication, and there's far more long-lasting effects that can occur. If you remember, during an action potential, when it begins, that's because of this slight opening of sodium ion channels that cause this little leakage in that triggers that threshold. That is often caused by the binding of neurotransmitters to these ionotropic receptors that cause those ion channels to open. To be specific, those are ligand-gated or mechanically gated ion channels, but for the purpose of this video, don't worry too much about what that means. Just remember that these ion channels open up, they're caused by neurotransmitters coming in, and that is what triggers the beginning of an action potential. One last important difference between chemical and electrical synapses is that chemical synapses can be either excitatory or inhibitory, while electrical synapses can only be excitatory. You're probably wondering what excitatory and inhibitory means, though. Those are just terms used to describe the postsynaptic potential, which is just another word for the action potential in this postsynaptic neuron. So as you can see in the electrical, you have the neuron that's the second one there, and the neuron in the chemical that's the second one here. When the action potential passes to that becomes your postsynaptic potential. An excitatory postsynaptic potential occurs when the sodium ion channels in the postsynaptic neuron open up and the likelihood of an action potential firing in that neuron increases. On the other hand, an inhibitory postsynaptic potential occurs when chloride ion channels open, and the likelihood of that postsynaptic neuron firing an action potential becomes less likely. This occurs because chloride ions are negatively charged, and when they rush into the neuron becomes more negatively charged, to the point that it's actually hyperpolarized, and that causes it to be less likely that an action potential will fire. And with that, we have reached the end of this series on Science with Jake, talking about neurons, action potentials, and synapses. Thank you for watching. I hope you learned something. I'm going to share this with the world, hopefully, probably, maybe not just for this project, because I think this is useful, and I hope that this can help someone who is studying for an exam, studying for anything, if someone that just wants to learn. I hope you enjoyed this video, enjoyed the series, and who knows, maybe there will be more in the future. Thanks for watching.